I looked at one species within these bacteria, Pseudomonas. This is a species we know differentially metabolizes atrazine. I tested all 160 samples with the primer-specific probe, and then I compared the two, and we can see that in the Pseudomonas of the bacteria within wasps habitually fed atrazine, they are able to metabolize more, as you can see by the dark streaks, compared to those of the control. So now let's summarize. Bacteria within wasps habitually fed atrazine had developed a stronger resistance to atrazine, and they could metabolize more of it compared to that of the control. This difference was seen in both random samples, and it was seen at the species level. So as we're working through this experiment, there's a couple of questions that we're going to be asking moving forward. The first is exactly how much atrazine can bacteria from each group survive exposure to. So far we've hit 600 ppm, but we haven't hit the limit yet. We want to see what the unknown bacteria are that can metabolize atrazine, so the ones that aren't Pseudomonas. And finally, we want to investigate the molecular basis for the degradation of atrazine across these bacteria. Maybe those within the atrazine-fed wasps are different than those within the control, and why? And answering these types of questions is going to enable us to have two implications. The first is bioremediation. So because atrazine is so commonly used across the US, it's been linked to polluting areas that have become dangerous to inhabitants. Specifically, in Wisconsin, there have become prohibition zones where residents in these areas have developed disease because they've used drinking water or they've been in areas that have a lot of atrazine. Bacteria like these, or the findings from it, would be able to restore these lands. Further, these bacteria metabolizing atrazine can help us with species that are currently endangered by exposure to it. I'd like to thank those in my lab who empowered me this summer to research and to learn about this project. Robert Brucker, Diane Schack, Guan Ongwing, Eric Hepler, and Brittany Birdie. I'd also like to thank those who have funded my research: the National Science Foundation, Harvard School of Engineering, the Rollin Institute, Boston College, and the Brucker Lab. I'm really grateful for the people who have enabled me to become a better scientist and have such a great experience this summer. Are there any questions?
Do you know what biochemical pathway atrazine is acting on to reduce the survivability of the bacteria in like the control group?
Yeah, so
Can you repeat the question, Eva?
Yeah, it's a good idea. So Chris asked, do we know what biochemical pathway is caused the bacteria, has caused the it to be toxic within certain bugs? So we have investigated, the other part of my lab has investigated how atrazine has had negative effects on the honeybee, and we've seen that certain byproducts of atrazine has looked at it. I haven't personally researched it as much, but what we've done is we've paid special attention to the pathways that enable these bacteria to break down the atrazine to begin with. So for the Pseudomonas example that I gave you, that actually uses a plasmid. And so we're going to be able to compare whether or not this plasmid has changed, whether or not it's by how much, or at the molecular level between the different groups. Does that help? Yes?
So why did you choose wasps?
That's a great question. So these wasps are special because they're parasitic. This means that they lay their eggs within what's called a puparium, or an undeveloped fly. When this wasp lays its eggs in the fly, then the eggs are able to be in a contained environment, so we don't have to worry about other environmental factors that impact the microbiome.
Could you use something else that parasitizes the victims?
Yes.
Besides wasps?
But another advantage to these wasps too is that they have a really short generation period, which has enabled us to see the effect after 73 generations. Another point that's really cool about these wasps is that they're haploid diploid. So while the male only has one pair the male is haploid, the female is diploid, and so we're able to select for mutations whether or not they're dominant or recessive between the genders.
You mentioned 72 generations. Is it that you have a host wasp that is exposed to atrazine, and then all subsequent wasps are also exposed to atrazine, or just the initial wasp is exposed and then all the subsequent wasps are just kept in the same environment as the control, essentially?
That's a good question. So to rephrase, is it that the wasps are exposed to atrazine at each generation, or that the first generation of wasps was exposed to atrazine? It's the former. The wasps, in the atrazine example that I gave, each generation was fed atrazine. And that enables us to look at differences not only in the 73rd generation, but maybe at the 40th generation or the 20th generation, because the impact of this is going to increase as more wasps are bred. Thank you.